Theodore Goodson III. He's the Richard Barry Bernstein Collegiate Professor of Chemistry and Macromolecular Science and Engineering at the University of Michigan. He's been recognized for his excellent research and has been awarded a Sloan Fellowship, is a AAAS and ACS Fellow, and a Senior Editor at the Journal of Physical Chemistry, among many, many other accolades. He's also been recognized for his excellent mentorship, which I always think is really important to add in there as well. He's done some really important work towards understanding dynamics and optical properties of organic materials and nanomaterials using ultrafast spectroscopy. And we'll hear a little bit about that today in his talk. So thank you very much.
So, okay, first of all, remind me, can you hear me? I don't want to start talking and like 30 minutes later you tell me, hey. Okay, thank you, Vanessa, and thank you, Marco and Rodrigo, as well as Lou, for inviting me to talk to you guys today. And thanks to everyone who's out in the audience in the virtual world who's listening. And I want to tell you about some of the work that we're doing in my lab that's extended over pretty much my entire career, and kind of give you about a sense of where we're moving with this kind of research. It's a very kind of important area of research that kind of touches on a number of different topics, and just wanted to kind of share that with you today. So this is a picture of our group, and they're all wearing their mask, and they're all very conscientious, excellent scientists. It was a group of a number of different disciplines, by the way, from analytical to physical chemistry to physics, applied physics, and polymer engineering. And so we have kind of a diverse group of scientists and engineers in our group, but we have one group meeting and everybody kind of learns something about each other's projects. So I'm going to tell you about, some of the work that we've done over the last so many years. And it actually it's related to solar. I mean, that's kind of like the motivation. So I've just put that out there. We're still grasping for that special material, whether it be organic or inorganic, that can do things like capture bigger parts of the spectrum, or have more efficient electron hole kind of capture and diffusion, or go through these different kinds of processes in a better way, electron hole relaxation, pair diffusion, charge transfer, and so on. And so, and extraction. So we're still looking for materials. When I started this work about 22 years ago or so, there's been a great deal of progress that's been made. For example, the efficiency, I would say, over that time has doubled for organic systems. That's just speaking roughly. When I started to get efficiencies that were 10%, that was a big deal, and that's not such a big deal anymore. And so 20, people expect close to 20%. There's lots of groups in China and so on that have published those kinds of things. So, anyway, so I think we want to talk about those kinds of things first, and then, and what's important to us is not necessarily to find the magical material, because we're not really making anything. It's just I'll throw that out there at the beginning. We didn't make the materials, we collaborate, and you'll see we have excellent collaborators who actually synthesize things, both in universities and in industry or at national labs. And our work is really to try to understand the mechanism of energy transport. And it actually matters. Actually, I could say that. And some of the folks that are here and listed in some of these references would say, that maybe 20 years ago, it wasn't clear if long-range exciton transport was really that important, for people to worry about for solar or for LEDs. And I can say now that people agree that it is important, okay? But I'm going to show you kind of like the journey that we went on, not just to prove that, energy transport in these systems matters, but actually that it can help us predict better materials. And, of course, people are still very interested in trying to capture, in essence, some of the mechanism of the natural light harvesting system in terms of its efficient energy transport mechanisms around circular aggregates, and then long range energy transport that happens in these systems as well. So, what are we interested in? So, well, this comes to the question of we're time resolved spectroscopists, nonlinear and time resolved, just like Vanessa's working on her kind of time resolved spectroscopy. We're working on systems, and we've decided to, very early on to look at artificial systems. Okay, so we're artificial people. We want to look at new materials, and our dream, one day is that someone would actually find the structure function relationships and the physics and try to understand this process such that we wouldn't try to make something like this, but we would take the essence of the physics out of this and make something that, somebody could make in buckets of like a polymer. Wouldn't that be, and, as they say, it would be as cheap as buying polystyrene, right? And so that's our dream, and we're sticking to it. And we haven't gotten there yet. But along the way, we have learned a lot of things about intermolecular coupling and energy migration. So this idea of coherence, which we're talking about a lot about today, is important in terms of spectroscopy and computing. It's important for energy transport and electron transport, as well as exciton effects and efficient transport. So this idea of coherent or wave packet motion matters. That's the first statement to think. It matters in practical things like solar cell materials and LEDs. And the investigations of these effects in organic systems, which is something that we've been doing for the last 20 years, can be helpful in understanding the properties of these materials as well as their performance. And so at a local level, we're interested in how these structures, quantum dots, aggregates, and how to probe these things. But at the end of the day, we're trying to understand this coherence, and we're using different tools to get at it, from quantum spectroscopy to ultra-fast spectroscopy. And I'm going to talk about that today. So we still have been interested in phase coherence, amplitude and phase coherence in these kinds of materials. And the reason why is because you can get something out of this. If you can actually find systems that have very strong amplitude and phase correlations, and you can measure that, you can get enhanced exciton effects, which could give you, enhanced light emitting materials. You could have enhanced charge transport. You could have magnetic type spin superposition. You could have coherent Raman scattering that could give orders of magnitude improvement. You could have quantum coherent in the super suppression and the superposition kind of light spectroscopy, which we've worked on. And you could have delocalized states, which, with less disorder. There's a huge advantage of having a coherent type process, wave packet motion in some novel organic system. It gives rise to these extra enhanced physical chemistry effects. And ultimately, they could give a rise to enhanced effects in practical applications like solar and LEDs. So coherence and excitation dynamics really, and from the standpoint of what we've been doing over the years, has been to look at energy transfer and the coherence of that energy transfer process, which relates to how fast the transfer is compared to the dephasing time or the T2 relaxation time, or just measuring the emission directly and looking for coherence in the emission, in which case you would look at the rate of decay time and compare that to the T2 relaxation time. Both of these are valid approaches to use in order to quantify or clarify the coherence in your system. And I'm going to talk about both of them today. So when we started, we didn't have all the instruments we have now. We did lots of fluorescence up conversion. And I still think that's a wonderful tool to use to investigate ultra-fast coherent processes, because you can use up conversion, the fluorescence decay measurement to look at decay times, less than a picosecond. And then you can use the anisotropy to look at energy migration in these systems, maybe even less than 100 femtoseconds. And you can do that in solution and film. We used that when early on, we used that up conversion. It was the workhorse. And I still say that you can learn a lot. There are a lot of more sophisticated, better techniques out there today, but I always try to make the point that we learned a lot from up conversion, using up conversion. And this is Megan Oren. She's a graduate student working on ultra-fast, mainly transient absorption as well as up conversion. And she's worked on looping used polymers. But we were doing a lot of ultra-fast, femtosecond spectroscopy, and at the beginning, there are certain ideas that happen, around, I was going to say about 20 years ago. It's actually exactly 20 years ago. One of them was that you could use these kinds of dendritic structures. This is for the case of enhanced solar applications. You could use these dendritic structures, could have these increased coherent domains. There were some synthetic as well as theoreticians who actually thought that you could have this energy funneling idea. That means that you would take, a small building block and make a dendrimer out of that, and then make a branch structure and make it second generation or third generation dendrimer after that. And the idea is that the absorbed photons the chromophores on the outside of the branches would energy transfer down to the next branch, and then of course delocalize inside the core, the energy funnel idea. And you could use fluorescence anisotropy to measure those kinds of things back in the day. And Mahinda was an excellent graduate student who did this at room temperature and as low as 4 Kelvin as well. And you could see this was a viable idea, that if you had these multi-chromophore systems back in those times, that you give this really fast energy transport mechanism. It was useful. It could suggest that your mechanism of energy transfer would be so fast that you could get more chromophore than what each chromophore would give you isolated. So those systems, by the way, were made at that time Yale University by John Hardwick, and then the colleagues in Japan, Kabuki and Ogawa, tried to create a system in which was very similar to the natural system, this ring-like structure, but it was synthetically, artificially made. And it's got similar kinds of chromophores. And the idea, again, is that we would actually see this energy transfer process, and the transport would be so fast that you couldn't explain it by some kind of hopping Forster kind of mechanism. And Jeff Raymond and Ajit Bhaskar worked on this system. And they used a variety of techniques like nonlinear spectroscopy, as well as two-photon spectroscopy, as well as fluorescence upconversion and transient absorption. And they found that that was actually the case in these structures. But one thing about many of these systems, I'm not going to go into every system we studied, is that the complexity of the structures were getting quite high. Remember, I was saying that we were looking for something that was fairly simple. The idea by Iota came, in which you would make these oligomers, long oligomers of thiophene with benzene rings. And then at each end of the oligomer would be a tethering point, and he could throw that into solution, and it would literally spontaneously form these rings. And so we kind of thought that's good, pretty much very straightforward approach, right? And what we found was that, again, the coupling between the chromophores, this coherent idea, it's kind of like the second slide I showed, that the transfer rate was enhanced by a factor of at least 300, suggesting that this coherence is so strong in this system going around that ring that you're seeing this kind of energy or quantum coherent kind of process, which gives you enhanced rate and then could be enhanced efficiency for the solar device. So, but the interest in making those rings, and actually the geometry of those structures, was not so clear. Luping Yu then went back to the drawing board and said, well, okay, so people started with polymers, because polymers is the best case scenario, by the way, because you don't worry so much about the regional specificity of the system. You don't worry about the molecular weight. You literally just live with the polymer. But that didn't work. So then people went to these oligomers, and then they went to dendrimers, then they went to these circular aggregates, and now they come back to the oligomers again. But these were the fused kind of structures And these fused structures have this kind of pi-pi stacking, which is favorable for this enhanced, again, charge transport in this system. And so Jeff Raymond and Ricardo Vasquez in our group studied these. And he was able to show that you could get this enhanced. And these are just linear molecules, not just, these are linear molecules, okay? And so, you can kind of understand the mechanism of the energy transport. All of this is to kind of tell you that you can have this kind of picture here where you say size, this is molecular weight, but I'll just say size. And this is a cross section. This is a measure of the transition moment to the fourth power, which is a figure of merit for this coherence, okay? And what this graph is trying to show you is that, of course, as you increase the size, you're getting that enhancement factor. But we're kind of interested in the intermediate size things, right? That are not so big and complex, but you still get very large enhancements. And we still were working at that. There were some people working at the Air Force and other places. They had chromophores. You can see they're very small. And then there was those thiophene rings are out here. So they're very good, but their size is large. And other structures. Here's those porphyrin ring structures are right there. So we're still kind of looking for that. And that's kind of all of those measurements I just told you about were done in solution. And the basic take-home message for that first part there is that, coherence matters. Finding structures preferably that you can kind of self-assemble from smaller building blocks into larger building blocks is a way to go. Having very large structures in which the orientation and the geometry matters is probably difficult, okay? So these are structure functional kinds of things. But that's just energy transport outright, okay? You're looking at the wave packet motion after excitation with femtosecond pulse. There are other mechanisms to enhance the performance of solar cells, and one of them is singlet exciton fission. And you've heard talks by Joseph or Mark Ratner and other people. This has been around for a while. But this topic of my talk today is really about coherence. And so you might say, well, how does this connect with coherence? So first of all, if you're not familiar, singlet fission is the idea that you have one singlet which can be basically distributed amongst two triplets, if you will, okay? And it's a quantum mechanical process. And what happens is that the singlet state here forms, before it forms those two triplet states and this fissionable process, it forms some kind of singlet state that has triplet character. How's that for a mouthful, right? And it's called the so-called double triplet state. And, it's a real coherent process of the formation between these two triplets forming this kind of strongly coherently coupled double triplet state. So much so that, folks like Chris Bardeen back in 2012 measured the, quantum coherent or beating between these two triplet states in a particular singlet fission molecule. You can actually see the ringing, if you will, of the interference between these two. Well, we have investigated the dynamics of singlet exciton fission and related processes, mainly from the standpoint of looking at these kind of coherent processes in these systems. And with that kind of work is still ongoing in my group. It's a result of coherent electronic coupling, which is important for this process. But I'm not going to talk about that as much, and only to say that a very excellent graduate student who will be graduating next Tuesday is Angela Mathiki, and she has worked on these PDI type singlet exciton fission materials, where she's gotten more than 124% triplets. And that idea of that coupling of those two the coupling of the two triplet states are the actual dynamics of the double triplet state, and the structure function relationships of actually making very small changes which affect the twisting and orientation of these PDIs around the central unit was the study that she did, trying to actually carry out a structure function relationship between that coupling of the double triplet state and the actual changing the geometry of the molecule very slightly. Again, these were made by Looping Yu in a different study. But what I did want to talk about a little bit was about some materials and some effects that we've been working with, and we've collaborated with LGO. And here is actually related to something called thermally activated delayed fluorescence. And Lloyd Fisher has been a person who's been working most recently on that. And Ricardo Vasquez was a graduate student that worked on it previously. The basic idea, of course, is that you would like to create more singlets, and there's from the deuterious triplets that are formed. The reason why is because people are interested in having longer lived devices, because the device efficiency is sometimes affected by triplet annihilation. And if you can reduce the numbers of triplets that are left in the system, and it goes back to singlets through this reverse intersystem crossing mechanism, then you could actually have a longer lived device and maybe even more efficient device. People like Adachi, as well as others, have investigated using different hosts, so you can energy transfer energy from the singlet of hosts to the triplet of other systems. But what we're trying to do is to develop, and this is what we did in collaboration with LG, is to develop a way to characterize this coupling between the triplet and singlet. Again, could this process be a coherent process? And here we go. So the first to investigate this was Ricardo. And we had developed a technique of using transient absorption measurements. Essentially, the rate and the quantum yield and the rate of the excited state absorption decay of the triplet state can be used as a figure of merit of the reverse intersystem crossing rate. And what we had found in collaboration with LG is that they had measured efficiencies with these devices, and we had measured with our approach this reverse intersystem crossing rate. And we saw this very strong correlation that, of course, those that had the higher rates of a reverse intersystem crossing, as measured from ultra-fast transient absorption, had larger external quantum efficiencies. And so this suggests that this could be a useful approach to kind of first quantifying the ability to see if a material is even useful. But secondly, we're really after, again, the structure function relationships in these systems. And so Lloyd has really been interested in collaborating with Dr. Lee in Korea. It's been really interested in looking at changing donor systems, making structure function relationships, and going back to the question of why. And so in a more recent study that just was published last week or 2 weeks ago, he investigated these carbazole kind of system, which had 2 donors and one had 1 donor. He measured the reverse intersystem crossing rates in these systems. And then he, in collaboration with Paul Zimmerman, who's been on so many papers with us. So Paul Zimmerman and his group, and particularly Hanji Jing, who's also going to be graduating this year, did excellent calculations to show how this idea of making these thermally activated delayed fluorescent chromophores is to have them as distinct as possible in the HOMO and LUMO, to have the excitation either localized at certain parts of the donor, and then in the HOMO, in the LUMO, and then in the HOMO to have it completely opposite. And so some of the systems with the carbazoles by Adachi really showed this really dichotomy of how different they are in the HOMO and LUMO, and how localized the system can be. So I'm kind of saying that we're still very interested in energy transfer and coherence in these processes. We can see that there's still some interesting materials around that we're interested in that have very fast energy transport. But at the same time, we're interested in other mechanisms that will enhance the efficiency too, such as single fission as well as TADF. So that's mostly done in solution. What about the question that lots of people would always ask is what happens in the case of things are done in the solid state? Or can your results completely be correlated to what happens in the solid state? And this was a question that people would always ask. And, you would come up with answers. But until we actually found a way to do similar kinds of ultra-fast measurements and measure coherence in the solid state, we were at a disadvantage. So we developed this femtosecond time resolved near field microscope in my laboratory. It's a 2-photon microscope that has really fast time resolution, and I'll show you about that. And it allowed us to really do things, not only from the standpoint of really kind of good physical chemistry, but also for something practical. Somebody would give you an actual device from LG and you just put it in the, just measure it, right? And you don't have to dissolve it, okay? And so, anyways, we had built this microscope. This was a collaboration between Taisoo Kim and Oleg Bornowski, who's a senior scientist in my lab. And let me just tell you a little bit about it. But actually, it gets its remnants from, of course, a graduate student who was thinking. And Dr. Naranga, who was an excellent graduate student here, he's now a professor back in Sri Lanka, was really trying to push this N2-photon INSUM harder. And so he had started already to look at how these processes could be done in the solid state. And he had worked on these tiny metal clusters, which I'm not going to have time to talk about today. But his research was really to try to quantify things in the solid state, not only just looking at their bulk properties, but single molecule kinds of properties and single molecule dynamics. And so taking that part of the piece, the spatial part of the piece, Taisoo and Oleg built this time resolved 2-photon near field optical microscope with time resolution of less than a femtosecond, less than one. So anyways, this system has this so-called phase lock stable pulses. And the reason why you can get this time resolution is you use this birefringent BBO wedge, which has the refractive index different in two different directions, which will cause a retardation of one of the light in one polarization from behind the other. And then now I have a delay that can be as short as 0.015 femtoseconds, but the reproducibility or repeatability is only about 0.2 femtoseconds. But you can have a delay line that can be as long as hundreds of picoseconds or as short as 100 or 200 femtoseconds. So this technique combined with the work that Naranga had been doing allowed us to do time resolved spectroscopy in a 2-photon near field optical microscope. That's a mouthful. So what are we going to do with it? So we were interested in looking at these kinds of time windows of coherence and wave packet motion in these systems. And basically this is going to be kind of like a, if you will, a photon echo experiment in a microscope, okay? And the first pulse is going to induce a coherent polarization in the system. And this coherent polarization will oscillate at the laser's frequency, okay? And then you send in a second pulse. And this one will also have a coherent polarization. And it's going to have an oscillation that's of the laser's frequency, but it's actually going to interfere with the first because you're not going to let the separation be too high. And this interference then will be kind of an indicator of the wave packet motion, because then we're going to read out the 2-photon fluorescence as a readout from this as a function of the separation of these two pulses, okay? And so, basically what's happening is you're going to end up with three kind of time windows. The first time window is going to be what happens right immediately after the laser fired, right? And so this so-called the optical interference area, this is within the autocorrelation function of the laser. And so this is right on top of the autocorrelation function. And this really just looks like the laser's oscillation. And then if you wait too long, it's a very long time between the red and the green, or the first and the second, then there is no interference anymore. And then you're just looking at the oscillation of the second pulse. This looks like the laser too, but it's at a longer time. Really, the science for what we are interested in happens in the middle. You would hope that there could be a polarization due to the first pulse and the second, some interference between these two. And here's now a molecule with that transition moment to the fourth power, and it's going to interfere with that. And that's going to be the dynamics that you want to investigate. And so that's the coherent polarization area. That's the hope, is that this is where the molecules regime will happen. And, the so-called wave packet motion, which is the difference between the autocorrelation function and the two-photon interferometric fluorescence signal from the substance, right? And it hopefully will last a long enough time that we can measure its coherent polarization, which is not taken for granted. So this idea of using this phase-locked coherent polarization idea is not new. Using it with a two-photon INSUM is, okay? So Van Holst, he had done this with a confocal microscope, and he actually did an ultralight harvesting system where he did see some kind of mode of wave packet motion associated with the natural light harvesting ring. And then Norbert Sherrod had done this also with SHG on looking at some silver nanoparticles. So we go back to looking at simple molecules who can have this kind of coherent energy transport, simple enough that, that you can understand the structure, and as well as people can make it, and you can make a device out of it. And so we were working with these PDI systems. These were made by Yuping Yu at the University of Chicago. We had done some theoretical collaboration with Paul Zimmerman's group, Kazito Madhu and Benedetto Carlotti were the principal people in our group that worked on these structures. But we had done all these kinds of experiments first in solution. And what we had found is that they have very interesting fast energy transport and large charge transfer properties, but it was in solution. And here's that same story again. Does it matter what happens when you put it in the solid state? So this is kind of what the data looks like when you do that experiment I was just talking to you. There's Tai-Soo Kim. And so you can get the spatial and the time resolution of this at the same time. And so you can see the lasers oscillating, and the signal, the two-photon signal, will oscillate, as you can see, with the laser at the laser's frequency. And then depending on where you actually investigate this, either in those 3 time windows, remember the very early stage or the very late stage is not so much of interest to us. It's that stage and that window, that time window in between. But you measure those odds. This is a very laborious kind of experiment. And so, people in those days used to work on the weekends. And so, people would actually measure these amplitudes, and he would actually find that if you make a plot here of the delay time versus the fast oscillation amplitude, you can see that in the case of these PDI dimers, you can see that there is an actual mode actually that's connected with the wave packet motion. And if you look at another point, so you can actually do this, and this is kind of useful because, with the case of a device or a material, you could have a film that looks perfect and it looks like it's very homogeneous, and you just go to different points on that device and film. The hope was that you could use this approach and actually see that even though it looks nice and smooth, the dynamics of energy transport are different at different regions based on the crystallinity or lack thereof, or amorphous, lots of reasons. But anyways, we found that. And then when you add the acceptors, you have the donor and the acceptor, you add the PTB7, then you can see a really interesting result that the PTB7 actually hides and engulfs the PDI chromophore system. And you can see that oscillate at the laser oscillation frequency. long story short, you can actually see that there are domains of energy transport in these systems. It's not a complete homogeneous process. Interesting. And Federica then said, well, if you guys are going to go back to polymers, in collaboration with a professor in China, Dr. Ma, we actually investigated these dendritic structures and showed that, there are also domains of structures and films there. But then, we really started to investigate the process of coherence in systems other than the organic small molecules and other than organic dendrimers and the macromolecules. And so we got interested in peroxides. And this was in collaboration with Emily Weiss as a part of a DOE kind of group grant we had with Mike Wazalewski and Shal Mukamel. And so I was kind of hesitant about looking at peroxides, cesium lead bromide. And Federica did a wonderful job on this project, not only on the experiment, but actually pointing out some of the interesting things about peroxides that are still not completely clear to us. And so one of them is actually just looking at the energy level diagram, which has this really interesting, well, it has the spin-over coupling, which you understand, but it has this so-called Rauschbach effect as well, in which you get a splitting, and this is actually the singlet state here. And this Rauschbach effect is going to play a role here in what we're going to talk about in terms of coherence. You already know that these peroxides are really useful. There had already been some reports of coherence in the emission of peroxides at low temperature with about 80 picosecond decay time at 5 Kelvin, and suggested that there might be some use for these photons emitted from such materials for single photon emitters. But the mechanism of this coherence and the range of it was still not clear to us, especially at room temperature. So Federica did a wonderful job of characterizing these structures for the first time in terms of their 2 photon. She did the normal things, looking at the 1 photon properties, but then she did the 2 photon properties as well. And so investigating these 2 photon properties, she saw that, these quantum dot structures have enormous 2 photon cross section, suggesting that they have this enhanced transition moment. Again, I keep coming back to that enhanced transition moment, which is connected with the second type of coherence. Remember this emission coherence. And so she was able to show that the cross section for the cesium lead bromide peroxide structure was, at least order of magnitude larger than for the other types of quantum dots that she studied, including cadmium selenide and others. She then measured up using up conversion. She then measured the fluorescence decay of these systems, okay? And the fluorescence decay was interesting, okay? It suggested that they have huge quantum yields, right? And that this lifetime is on the order of hundreds of picoseconds, okay, at room temperature. And so, all of this is kind of pointing towards some kind of, coherent process. And then she tried the power dependence and small power dependence, which could have suggested that there's a contribution from some kind of Auger or hole filling process involved in the emission itself. So then in doing this 2 photon time resolved near field optical microscopy, she really learned how to make really good films. That was a really challenging part of this project, is making reproducible films so that we could look at a single quantum dot. And so we start out from big to go to small. And actually the diameter that the AFM part of the 2 photon near field optical microscope time resolved optical microscope showed agreed with the TEM kind of results on the order of 10 to 12 to 13 nanometers, depending on the sample. So what she did was then to investigate the time resolved properties of these quantum dots, just as the same way that Taisu and Oleg had done with the organic system. And I should remember, this was actually during the pandemic. Over the pandemic time, one of the pieces of the equipment actually fried and it was left frying for like a week. And so Federica had to find, ways to fix this piece of equipment when we had hardly an electronics shop. We had no electronics shop here. And so she used her independence, her forthright kind of go get it done kind of ability to actually find a place that actually fixed it for us. And then we got this experiment running, and she was able to get this beautiful data, which showed that the peroxides, quantum dots, have a decay time of about 150 femtoseconds, which is this exciton coherence time. This is a long time when you think about it in comparison to gallium arsenide or bismuth or calcium selenide. And so the question is, and this is the comparison. So later for this paper, she did the cadmium selenide as well. So this suggests then that, okay, you can use this approach to look at that other kind of coherence, looking at the coherence and the emission decay of this excitation of these quantum dots. And, this long coherence time must have something to do with these low-lying triplet states in these systems. But how this affects, those triplet states must have some kind of orthogonal orientation to each other, and how they're strongly coupled in this kind of coherent mechanism is still a question mark to me and to us. But it certainly as at room temperature has shown to be something unique in comparison to other quantum dots. Okay, so I don't have time to talk much about these metal clusters, even though the title of my talk was Organic and Inorganic. I just wanted to say that we're still very interested in looking at metal clusters, and from the standpoint of understanding structure, bonding, understanding excitations in these systems as a result of the structure and bonding, and then of course, their applications. And so we have studied gold and silver and in mixed clusters, and we will continue to study these things in collaboration with the Armor Research Laboratory. But it's just didn't have enough time to discuss this today. If you want to talk about this, or if anybody is interested, please, you can shoot me an email. So the final thing I wanted to mention that's in my laboratory is that I've been talking about using ultrafast spectroscopy to investigate coherent excitations in organic systems. And basically you're using things like upconversion and transient absorption, photon echo, and this ultrafast microscopy to probe the process of wave packet motion. There's another tool that we've been using, which I think is equally as powerful and is starting to come into fruition, is the use of non-classical states of light. And in this case, you're using the high degree of correlation, which could give you enhancement of this coupling between the light and matter. And that could result in higher selectivity and sensitivity. And, there has been some work experimentally and theoretically, not only in the physics regime, but also in chemistry. There's a lot to be learned. There's lots to be, standardized as well as just to be understood. But I think there's a lot of activity that wasn't there when we started on this 20 something years ago. And the power of the use of non-classical states of light is that you could transfer the entanglement between field matter, and you could create so-called new probe new material states, right? And this is something that you can hardly achieve with classical light. And people like Shao and others are thinking about new dimensions in spectroscopy, where quantum interferometry is something that we've been working on, and others, as well as entangled nonlinear spectroscopy. And then, of course, the new molecular properties can be activated through the virtual state kind of selectivity. It sounds like we're pretty far off from that still, but at least now we're starting to think about, how that might happen. And due to the high correlations, of course, the noise characteristics can be diminished, and that's something that we're always interested in. So in my group, we have developed an entangled photon microscope for biological imaging. And we already have published that in terms of looking at small nanocrystals, but now We have a paper out that's coming out in terms of looking at cancer cells. We've worked on this quantum interferometry. In this case, the so-called Hung-O Mandel interferometer, and we worked with Shao on that. But you know, a lot of the work has been involving trying to come up with some kind of structure function relationships in terms of entangled two photon spectroscopy. While we're actually doing more of the interferometry and microscopy these days, that's kind of how it started. Ryan also, and Juan Villabona, who's in my group, and Ryan Burdick, who's now at the Air Force Research Laboratory, you know, we're very interested in the importance of entanglement time. And in working with Mike Wazalewski, how can we transduce entanglement from photons to spin? And then how can you utilize that? And is the last area. Maybe you could utilize that in investigating things like biological processes. So there's a lot of room for involvement. There's a lot of room for discovery still, but you know, I kind of started this talk about talking about what was around when I started, and those ideas about coherence and energy transport were already there, but the instruments and the spectroscopy got better, and we developed things like, you know, ultrafast microscopy. But now we have something totally new, you know, using non-classical light to answer the same question, and so I expect to, you know, to see some more growth in that area. So now it is about, I think about 50 minutes, so hopefully we have some time for questions. Is that right, Vanessa? Okay, and so I think I've mentioned everyone through the time who have collaborated. Of course, through the years, you know, I've collaborated with Lu Ping Yu, and you've probably seen a lot of papers with Paul Zimmerman on it, yeah, and George Schatz, and we're doing the singlet fission, Juan Casado. And I tried to mention all the students when I was talking about them. And we're grateful for all these agencies over the years who have supported this research that I've talked about today. And this is a picture from last summer. This is a long time ago. No, this is about maybe 5 or 6 years ago, I guess. And then this was just 2 or 3 weeks ago. And I thank you for your attention.
Thank you so much. Thank you so much for the wonderful talk. That's just a wonderful overview of a very large body of research, very large, very interesting body of research. Thank you very much. Just quickly, before we get to questions, I just want to mention a couple of things. I want to say thank you to our sponsors. So thank you to Phase Tech and the ACS PHYS division for sponsoring the webinar. I also want to mention that there'll be an opportunity for grad students and postdocs to stay on and ask questions after, and we'll have everybody else leave in case you'd like to do that as well. And since, you know, it's not a completely overwhelming number of people, we might be able to allow people to unmute if they have questions. So if you would like to raise your hand, we can go ahead and unmute you. But I'll start us off with a question. I think Marco has a question as well, but I'll go ahead and start us off with a question. So one of the things I'm often interested in, especially going from a solution phase to sort of like a solid state type phase, is really the role of disorder, particularly intermediate regimes of disorder. And it seems like, especially with the new capabilities that you're developing, you really have a way to get at, you know, the role of disorder, different kinds of disorder, and how it works in different systems. And I was wondering if you could comment on that a bit.
Right. And somebody like LG, they're very sensitive to that because and so you and I think of disorder, we probably try to do something obvious, right? Yeah. Give me a completely crystalline material and give me a completely disordered material. These people, they are looking at something to us looks like it's ordered, but just a tiny bit of disorder makes a huge difference that we normally wouldn't be able to tell, okay? Just from looking at like doing, if you just measure the film thickness or you do AFM, you would just, it looks good, you know, right? And so they're always looking for a technique that's not just a visual technique, like will give you some kind of physical parameter. And I would say using this microscope approach, time resolved microscope approach, that looking at that decay time could be that parameter. And to prove that it matters is huge, right? I mean, because it's not that you just want to do a bunch of ultra fast measurements, right? It says that the dynamics of electron hole motion in that device is sensitive to that order, that level of disorder, which is only a very small amount, which can only be detected by ultrafast.
Thank you. Marco, would you like to go ahead and ask your question?
Sure. I had actually a similar question to Vanessa's, but from the point of view of simulations, because I assume that most of these simulations are done on isolated molecules on these large chromophores. So I was wondering if you or your collaborators have looked at how the structure of these chromophores changes when you put it into the solid state, and if you can correlate that into changes in the spectroscopy.
No, I'm sure that there's still, like, you know, the classical kind of solvated thing, right? That would be great, right? You know, if you could correlate the change in fluorescence yield, the change in energy level spacing, and any of these kinds of parameters, that would be great. It's not typically done. I mean, so, well, okay, in the defense of the theorists, I should say, they're usually looking for trends, right? So we have, look at this graph here. There's 5 molecules. The trend, did they get the trend right? That's all, you know, right? And they usually do, okay? But, you know, one trend that you're asking about could be that I have a molecule and I put it into a rigid host or some kind of system of varying stiffness or something. And, you know, how does that affect? So I have not personally done, people have worked on things like that. I have not done it, but I don't think anyone has done that in connection with, you know, these kinds of ultra-fast measurements and the theory, you know, that somebody like, you know, Paul Zimmerman or George Schatz or somebody who can calculate the states for these systems, not to that level, right? That's a great question.
Are there any more questions from the audience? Please go ahead and raise your hand if so. Okay, if there are no more questions, let's thank Professor Goodson again. Thank you so much. All right, so grad students and postdocs, if you'd like to stay on after, you have a chance to talk with Professor Goodson. Okay, and we'll say goodbye to all of the other more senior members of the audience. Thank you so much.
Bye, Greg.